made in understanding amyotrophic lateral sclerosis, ALS. Then Sophie Yee examines new research into why eye muscles tend to last longer than other motor neurons in ALS and what this may mean for treatment. Also, in this collection, we look at the prevailing theories on the origins and the current therapies for multiple sclerosis, MS, and Huntington's disease. The cause of MS remains unknown, but new evidence has opened up new lines of inquiry. In one piece, we look at why researchers are turning to gray matter, as opposed to white matter, as the starting point for the disease. In another, researchers examine the autoimmune theory, which speculates that the myelin degradation in MS results from the immune system turning on itself. Recent studies suggest the aberrant response begins in the gut. Even though the definitive roots of neurodegenerative diseases are unknown, there are specific occurrences that link them all. These include genetic mutations, protein misfolding in neurons, and intercellular mechanical dysfunction. We wrap up this collection by looking at current preventative measures and treatments that target not only disease pathology, but also lifestyle changes as well. In A Rare Success Against Alzheimer's, the results of a large-scale Finnish study provide evidence that choices such as diet and exercise can help prevent cognitive decline. Although this news is far from a cure, forward movement against Alzheimer's and neurodegenerative disease in general is reason for optimism. As research and evidence accumulates, we get ever closer to curative therapies that can halt the debilitation and death of neurons. Karen Tucker, Book Editor. Section 1. Dementia. Seeds of Dementia by Larry C. Walker and Matthias Euchre. Under a microscope, a pathologist searching through the damaged nerve cells in a brain tissue sample from a patient who has died of Alzheimer's disease can make out strange clumps of material. They consist of proteins that clearly do not belong there. Where did they come from? And why are there so many of them? And, most important, what do they have to do with this devastating and incurable disorder? The search for answers has turned up a startling discovery. The clumped proteins in Alzheimer's and other major neurodegenerative diseases behave very much like prions, the toxic proteins that destroy the brain in mad cow disease. Prions are misshapen, yet durable versions of proteins normally present in nerve cells that cause like proteins to misfold and clump together, starting a chain reaction that eventually consumes entire brain regions. In the past 10 years, scientists have learned that such a process may be at work not only in mad cow and other exotic diseases, but also in major neurodegenerative disorders, including Alzheimer's, Parkinson's, amyotrophic lateral sclerosis, also known as ALS or Lou Gehrig's disease, and the concussion-related dementia of football players and boxers. Alzheimer's and Parkinson's, all evidence suggests, are not contagious like mad cow or, for that matter, the flu. Rather, the significance of these recent findings is that they provide scientists with a prime suspect for a slew of devastating brain disorders, a signpost that points toward a pathway for eventual treatments. Drugs developed for Alzheimer's might be used directly, or else inspire new pharmacology for Parkinson's, traumatic brain injury, or other terrifying conditions that rob an individual of a basic sense of self. Good news for tens of millions worldwide who suffer from neurodegenerative disorders. The new thinking owes a debt to research that first led to the discovery of prions. It began in the early 18th century with reports of a curious, fatal disease of sheep called scrapie, so named because affected animals compulsively rubbed the wool from their skin. Later, as scientists began to investigate the disease, they noticed under the microscope that the nervous system was shot through with holes. In the 1930s, French and British researchers determined that scrapie could be transmitted from one sheep to another. But the infectious agent was elusive and behaved strangely. The incubation time between exposure and symptoms was much longer than for conventional disease-causing agents such as